Hi, my name is Helena and I just completed my PhD at the Instituto de Medicina Molecular, supervised by Edgar Gomes. When you think of a cell, something like this could come to your mind: a rounded-shaped cell with one nucleus in the center. Well, skeletal muscle cells are very different. First of all, there's the size. Skeletal muscle cells, or myofibers, span from one tip of the muscle to the other, so each cell is the same in length as the muscle it makes part of, and its shape recalls the two. Myofibers are formed by the fusion of up to hundreds of smaller cells. So, instead of one nucleus in the center, myofibers have hundreds of nuclei that are well distributed in the cell. In fact, nuclei are so well distributed that scientists already show this could not be achieved randomly. Over the last few years, I have been interested in this subject. Why are the nuclei in myofibers so well distributed? One of the ideas that muscle experts have explored for a while is that nuclei are distributed well because each one is responsible for the area around it. If the nuclei are not well distributed, some areas of the cell would lack the appropriate amount of products from nuclei, such as mRNAs, and this could affect muscle function. According to this hypothesis, mRNA from each nucleus should be around the nucleus of origin. This occurs, for example, in the neuromuscular junction, the place where the motor neuron contacts with the muscle cell. In my work, I wanted to understand if this also occurs in all nuclei in other regions of the cell. So, I studied distribution of around 20 mRNAs that codify important proteins. We saw that a lot of mRNAs are enriched near the nucleus, which is in line with this hypothesis, but we discovered a group of mRNAs that are equally spread in the cell, regardless of the position of the nuclei. We tried to understand why these mRNAs are spread, while others are enriched around nuclei. Curiously, we found that the mRNAs that are spread in the cell are very large. So, we decided to test if size of mRNAs can contribute to their distribution by artificially expressing mRNAs with different sizes in muscle cells, and we showed that size does affect their distribution. Large mRNAs are more spread. We also saw that all mRNAs require an intact microtubule cytoskeleton to be dispersed away from the nucleus. The cytoplasm of muscle cells is highly packed by the units that allow the muscle to contract. So, the distribution of mRNA and proteins in these cells could be more challenging in general, especially for large mRNAs, which, as we discovered, are actually more efficiently distributed. So, there are mechanisms in place to efficiently distribute mRNAs with special characteristics such as a large size. This knowledge on biology of muscle cells can now be applied to understand how the position of nuclei can affect muscle function. Thank you.